Alright, so we're going to start talking about cellular respiration. Alright, so I'm going to review a couple of topics from Chapter 8 that really tie in with this material. Alright, so to start off with, we're going to talk about the carbon cycle. Alright, so cellular respiration is a component of the carbon cycle. Alright, so this kind of gives you a big picture view of how everything is integrated. So looking at the molecular side of it, the biochemical side of it, the organismal side, the ecological side of it.
Could a carbon atom that was once part of Albert Einstein be in you? All living things need to take in carbon-containing molecules to fuel their activity and build their cells. This is true for busy students and for all organisms. Let's look at how carbon atoms move through the carbon cycle in the Arctic tundra. Carbon atoms from carbon dioxide are incorporated into living things by producers, such as this plant. Carbon dioxide enters a leaf, moves into a cell, and goes into a chloroplast. There, in the process of photosynthesis, carbon dioxide is used to make sugar molecules. The sugars can be used to build larger carbon-containing molecules, carbohydrates, lipids, proteins, and nucleic acids. In this way, carbon that was in the air is now part of the plant. When a plant is eaten by an herbivore, or primary consumer, carbon in plant tissue enters the primary consumer's digestive system. There, plant molecules are broken down. These small molecules are then used by the primary consumer to make larger molecules for building its own tissues. In this way, some of the carbon that was in a plant becomes carbon in a primary consumer. Similarly, when a primary consumer is eaten by a higher level consumer, some of the carbon in the primary consumer is incorporated into the higher level consumer. Carbon-containing molecules in dead plant material, animal feces, and dead organisms are taken up by decomposers, such as fungi and bacteria. Decomposers break down these materials and incorporate some of the carbon atoms into their own bodies. We have now seen how carbon moves from carbon dioxide to producers, to primary consumers, to higher-level consumers, and to decomposers. How does carbon get back to the atmosphere? During cellular respiration, carbon-containing molecules are broken down, and ATP is made. Carbon dioxide is released as a byproduct and returned to the atmosphere by producers, consumers, and decomposers. Overall, this return of carbon dioxide by cellular respiration is closely balanced by its removal from the atmosphere through photosynthesis. The global carbon cycle involves living things on land and in rivers, lakes, and oceans. And by the way, considering the huge number of carbon atoms in each person and how quickly carbon atoms move through the carbon cycle, there is almost certainly a little of Einstein in each of us.
Alright, so early on in the semester we talked about the flow of energy within an ecosystem. Alright, so we talked about energy moving from sunlight from your producers to your consumers and to your decomposers. Alright, well, in this case, your producers take in sunlight, they fix CO2, which is, again, inorganic molecule, and they use water, the energy from the sun, to make glucose, sugars, and they produce, along with that, oxygen. Alright, the cells of the plant will then use that sugar and oxygen in a process of cellular respiration. The byproducts of cellular respiration, CO2 and water, along with ATP. Now you have a consumer come along that feeds on a plant or feeds on another heterotroph that's been eating the plant. The energy in the food gets broken down, and the mitochondria converts that energy stored in the chemical bonds of the molecules that make up the proteins, fat, and carbohydrates of the animal, and uses it to make ATP. Again, the byproduct of this is CO2 and water. Alright, so the oxygen that we breathe in from the atmosphere is used for aerobic cellular respiration. The CO2 that we expel, which was originally a component of organic molecules made by producers, or were organic molecules found in other living organisms that we've eaten, is released as CO2. That CO2 is then used by plants, fixed into an organic molecule like glucose. Alright, so it's one big cyclical process. Alright, so cellular respiration. Cellular respiration is all about producing ATP. Alright, and we're going to refer to ATP as the energy currency of the cell. Alright, because there are many processes in a cell or an organism that require an expenditure, cost of energy. And that cost is in the form of usually adenosine triphosphate, or ATP. Alright, so the hydrolysis of ATP yields a net release of energy that can be used by the cell to drive some biological process. Alright, so we're going to talk about three major biological processes. Alright, mainly we'll discuss aerobic cellular respiration. Alright, the use of organic molecules and oxygen to yield ATP. Alright, we'll talk about anaerobic respiration very briefly. Alright, it's very similar to aerobic respiration. There's a few changes we'll talk about. And then there's fermentation, which occurs in the absence of oxygen. All three of these processes are catabolic processes that yield ATP. Alright, so they are exergonic. Alright, so this is why we spent time going over exergonic and endergonic processes, over catabolic and anabolic, over potential energy, talking about Gibbs free energy. Alright, all this is going to come back around. Alright, we're going to start applying those terms to the process of cellular respiration. Right, and one of the other important things you're going to have to recall is oxidation and reduction, or redox reactions. Alright, so bringing up this issue of redox reactions. Alright, so I've already talked about this previously, but to reiterate the point. All right. A redox reaction involves one molecule being oxidized and another molecule being reduced. All right. So if in a chemical process one reactant molecule is oxidized, the other is going to be reduced. Okay. If a molecule is giving up electrons or hydrogen atoms, it's becoming oxidized. If a molecule is gaining electrons or hydrogen atoms, it's becoming reduced. Now, the molecule that is giving up the electrons, right, the electron donor in this case, is referred to as the reducing agent. The molecule that is accepting the electrons or hydrogen atoms is the oxidizing agent. Alright, so tying these terms of oxidation and reduction to the reaction of cellular respiration. Alright, so cellular respiration is basically a combustion reaction. You take a molecule like glucose, you combine it with the presence of oxygen, and you release energy. You yield CO2 and water as byproducts. Alright, so in this case, looking at what's oxidized. Alright, so look at glucose. Glucose is a nice little organic molecule. Get carbon, get hydrogen, get oxygen. Alright, you have, in this case, carbon bonds and carbon-hydrogen bonds, which again are non-polar covalent bonds. So in this case, there is a high degree of potential energy stored in those bonds. Alright, so when those bonds are broken, glucose is going to give up its electrons. It's going to give up the electrons that it's sharing with other carbon atoms and other hydrogen atoms. And these electrons are donated to what we call electron carriers. Alright, so these electron carriers are temporarily holding on to this potential energy that we have so far extracted from molecule of glucose. Alright, so when glucose loses the hydrogen atoms and loses the other covalent bonds holding it together, you end up with 6 molecules of carbon dioxide. Alright, so glucose is initially reduced and is oxidized, alright, to CO2. Notice, look at the balanced chemical reaction. Glucose, you have one molecule of glucose with 6 carbon atoms. Look here, you have 6 molecules of carbon dioxide. So the carbon atoms from glucose are the same carbon atoms that are released as CO2. All right. Oxygen. Oxygen is initially oxidized, and it's going to be reduced to water. So oxygen will eventually, at the end of this process, accept electrons, which were the original electrons from glucose. Those same electrons get transferred to oxygen to yield water. Alright, so oxygen gets reduced to water. Alright, so look at this process. Alright, glucose initially is reduced. Oxygen is oxidized. Alright, well, in this whole chemical reaction, look what happens. The glucose is initially reduced, is then oxidized to CO2. Oxygen is initially oxidized, is reduced to water. Alright, so a redox reaction, you're always coupling oxidation and reduction. And all this stored potential energy is then used to make ATP. Alright, so we're going to break down this process of cell respiration. We're going to talk about how glucose is oxidized to CO2. We're going to talk about how oxygen is reduced to water. And then lastly, we'll talk about how ATP is made in this process. Alright, so looking at our electron carrier molecules. Alright, so you have NAD plus and FAD. These are our little electron carriers. Think of them as little bitty taxi cabs. Alright, their job is to pick up electrons and they will shuttle them to the mitochondrial membrane. Okay. So they basically take electrons, potential energy, from various intermediates of cellular respiration and transfer that stored potential energy of electrons to the mitochondrial membrane. Alright, so here we have an example of a molecule that has stored potential energy. Alright. And it's going to become oxidized. Alright, so it's going to give up some of its potential energy. Alright, some of these electrons, some of these hydrogen atoms. And these electrons get transferred to NAD+. Alright, so this molecule initially is reduced, and NAD+ is initially oxidized. Alright, so when this molecule which is initially reduced is oxidized, it gives up its electrons to NAD+. NAD+ accepts the electrons, and it becomes reduced. Alright, now when it's reduced, it's referred to as NADH. Alright, so now NADH shuttles the electrons to the mitochondrial membrane, mitochondrial inner membrane. NADH, which is reduced here, gives up its electrons to proteins in the membrane. NADH becomes oxidized to NAD+, and the protein in the inner membrane becomes reduced. And then, through a series of redox reactions in the mitochondrial inner membrane, you end up producing ATP, and eventually, at the end of the electron transport chain, The electrons you initially extracted from this molecule over here, right, like glucose for instance, are going to be transferred to oxygen, which is one of our initial reactants in cell respiration, to form water. All right, so here oxygen is reduced to water.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen consists of a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, the products separate from the enzyme and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
Alright, so looking at the process of cell respiration. Alright, so keeping in mind this is a nice little combustion reaction. If you were to look at the entire process as a whole, and if you were to go from glucose directly to skipping all these intermediate steps, all right, giving oxygen the electrons to form water and produce ATP, you would end up something like this. All right, so it's kind of like burning sugar, burning wood. It's a combustion reaction. You combine sugar, combine the oxygen, you burn it, you release a tremendous amount of energy, a huge amount of energy. All right, you release CO2 and water. Now, this huge loss of energy is mainly in form of light and heat. All right, not a lot of it's very usable in that case. All right, so what happens in the body is our cells, in order to extract the energy it needs, it breaks up the process over very small incremental steps. All right, so it's a rather slow process in that sense. All right, you don't go from point A to point B. All right, there's a lot of small intermediate steps in between, so that you can extract and yield as much ATP as possible and reduce the amount of loss of energy in the form of heat. All right, so you guys looked at various chemical reactions involving a handout I gave you in class. And you went through and categorized these reactions as exergonic, endergonic, anabolic, catabolic. You calculated the delta G value, all right, that gives free energy. And that told you whether or not a reaction was spontaneous or non-spontaneous, whether it was exergonic or endergonic. And even when you adjusted the temperature from 20 degrees Celsius to 37 degrees Celsius, a reaction that was endergonic was still endergonic even at 37 degrees Celsius. All right, it would require a tremendous amount of heat in terms of temperature to allow that reaction to become exergonic. But it's not conducive to the life of our cells that exist at 37 degrees Celsius in terms of their optimum temperature, in terms of optimum functionality of enzymes. Alright, so this is why we have enzymes. Alright, the enzymes speed up the chemical process, but it also allows these biochemical processes to occur along with the utilization of ATP to allow reactions that are typically endergonic to become exergonic if you add a little bit of extra energy into the process using ATP. Because whether you're talking about a exergonic or endergonic process, both processes require some kind of energy input. You still have to get past the activation energy that a chemical reaction needs to take place. Alright, so looking at the different stages of cellular respiration. Alright, so in terms of a sequence of steps, you have the first being glycolysis. Alright, so glycolysis involves the conversion of glucose to pyruvate. Alright, so this takes place in the cytosol of the cell. The next phase, actually the next three phases or stages of respiration, all occur somewhere in the mitochondria. Alright, so in this case, the next step would be the pyruvate oxidation, right, where you convert pyruvate to acetyl-CoA. Alright, so this will take place in the fluid-filled compartment of the mitochondria that we refer to as the matrix. Alright, the next stage, you take the acetyl-CoA and it's sent through a process referred to as the citric acid cycle or the Krebs cycle. Now this also takes place in the matrix of the mitochondria. The last and final step is referred to as oxidative phosphorylation. So this involves the proteins that make up the electron transport chain in the inner membrane of the mitochondria. Alright, specifically these enfoldings that we refer to as cristae of the inner membrane. Alright, so going over the first step of cell respiration, glycolysis. Alright, so the name, glyco, sugar, and lysis. Alright, so you're splitting glucose. Alright, that's what you're doing in this process. You're taking a 6-carbon molecule and splitting it in half. Alright, so you end up with 2 3-carbon molecules. Alright, so this particular metabolic process is universal. Alright, so this is probably one of the oldest metabolic processes. Alright, because it's found in virtually almost every single living organism on this planet. Alright, it's both a aerobic and an anaerobic process. So that means it can take place in the presence of oxygen and in the absence of oxygen. Alright, so this particular process is divided into 2 main phases. Alright, we'll focus on the energy capturing and the energy harvesting phases. Alright, so kind of give you an overview of glycolysis.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
All right, so looking at this process of glycolysis, so we have an energy investment and an energy harvesting phase. So, glucose is brought into the cell, and initially this glucose is going to be phosphorylated by ATP. Alright, so glucose has the attachment of a phosphate group added onto it to 6-carbon. Alright, so here your starting materials are glucose, ATP, NAD+, ADP, and inorganic phosphate. Alright, so the ATP that you're initially using, alright, you're basically adding more potential energy to the molecule. You're actually, in this process of adding phosphates to it, you're making it a much more unstable molecule. You're increasing the amount of potential energy and making it a more unstable molecule so that it will eventually, through this process of phosphorylating various intermediates, it'll split apart. Alright, so you have this fructose 1,6-bisphosphate molecule that splits into 2 3-carbon molecules of G3P. Alright, so during the energy harvesting phase, alright, this is where we're actually capturing energy from various intermediates of glucose during this process of glycolysis. Alright, so now we have G3P, G3P, or glyceraldehyde 3-phosphate. Alright, so these 2 G3P molecules, alright, they're going to have electrons that are transferred from the G3P molecule to our little bitty electron carrier, NAD+. Alright, so NAD+ picks up electrons from G3P, and the G3P molecule becomes, in this case, oxidized. And the NADP plus molecule, when it accepts the electrons, it becomes NADH, and it's reduced. Alright, so again, you have a process of oxidation and reduction. Next, you end up with this intermediate 1,3-bisphosphoglycerate. Alright, now you still have 2 high energy phosphate groups attached. Alright, hence the name 1,3-bisphospho. Alright, so you have these 2 high energy phosphate groups. This molecule of 1,3-bisphosphoglycerate, alright, you have the transfer of one of the phosphate groups to ADP. Alright, so as a result, you end up with ATP. Alright, this process occurs again when phosphoenol pyruvate, PEP, transfers its phosphate group to ADP to form ATP. And you end up with pyruvate. Alright, so this process of glycolysis, what you end up with is a total yield of 4-ATP. Alright, because here you take glucose, alright, and you split it into 2 G3P molecules. Alright, each of those G3P molecules is going to be converted to pyruvate. And in that process, you produce 1 NADH and 2 ATPs each. Alright, so out of this you get NADH and 2 ATP. Alright, again, out of this you get 1 NADH and 2 ATP. So you get a total yield of 4 ATP molecules. Alright, but the initial process required 2 ATP molecules to be expended. Alright, so you have a net yield of 2 ATP, right? Because you invested 2 to start the whole process. You end up with 2 pyruvate molecules and 2 NADH molecules. Alright, so this is kind of simplifying the process. The energy investment phase. Glucose is going to have energy, ATP, that's added to it that makes it a little bit less stable. Alright. And then you end up with this G3P molecule in this intermediate phase. It's kind of transition between the energy investment and the energy payoff or harvesting phase. Alright, so now this G3P is converted to pyruvate, where you transfer the phosphate groups to ADP to form ATP, and you transfer electrons to NAD plus to form NADH. Alright, so how exactly is ATP being generated in glycolysis? Well, to explain this, we have a process called substrate-level phosphorylation. Alright, so you have some enzyme, you have a substrate molecule with a phosphate group attached, you have a molecule of ADP. Alright, both these bind to their respective binding sites, active sites of the enzyme. And you have the transfer of the phosphate group from the substrate to ADP, which now becomes ATP. Alright, so you have substrate-level phosphorylation. Alright, to look at this a little bit more specifically, alright, the conversion of PEP to pyruvate. Alright, you have this substrate-level phosphorylation. Alright, so here you have the phosphate group, phosphoenol pyruvate. Alright, this is a highly unstable bond. Alright, and it doesn't take a lot of energy to break this particular bond. Alright, so this bond is going to be broken and transferred to ADP. Alright, so as a result, you end up with pyruvate and ATP. Alright, the enzyme involved is a kinase enzyme. All right, so in this case, a phosphorylation reaction, the transfer of a phosphate group from one molecule to another, involves a kinase enzyme. In this case, pyruvate kinase, because pyruvate is the product of this biochemical reaction. And kinase is telling you that somewhere in this process, you're transferring a phosphate group from one molecule to ADP, or some molecule is going to be phosphorylated at the very least.